Genetic replication is essential for life. In this video, we will explore how DNA becomes RNA and eventually protein through the processes of transcription and translation. Protein synthesis has its roots in the central dogma, which states that protein synthesis requires two steps. They are transcription and translation. Let's begin with a brief overview. The first process is transcription, the synthesis of RNA from a DNA template. In this process, only one strand of DNA is copied. A single gene may be transcribed thousands of times. After transcription, the DNA strands rejoin, allowing for the process to replicate countless times. Some of the RNA produced by transcription is not used for protein synthesis. These RNA molecules have other functions in the cell. In between the two processes, splicing and expression keep synthesis going. A single gene is capable of producing more than one different polypeptide by removing different introns from the primary RNA transcript. For example, humans have an estimated 20,000 genes. These genes produce as many as 100,000 different proteins due to alternative RNA splicing. The second process is translation, the process where ribosomes synthesize proteins using the mature RNA transcript produced during transcription. It's also important to understand the different types of RNA involved in these processes. RNA has a variety of different functions in the cell. There are four types of important RNA. Messenger RNA, also known as mRNA, contains genetic information from DNA. It is a copy of a portion of the DNA. It carries genetic information from the gene out of the nucleus and into the cytoplasm of the cell, where it is translated to produce protein. Ribosomal RNA, also known as rRNA, is a structural component of the ribosomes. It does not contain a genetic message. Transfer RNA, also known as tRNA, functions to transport amino acids to the ribosomes during protein synthesis. Finally, small nuclear RNA, also known as snRNA, are strands of RNA complexed with protein producing small nuclear ribonucleoproteins. Now, let's go into more detail regarding transcription.
The enzyme RNA polymerase is responsible for creating RNA by copying the template strand of DNA. It pries the DNA apart and hooks together the RNA nucleotides. The RNA is complementary to the DNA template strand. RNA synthesis follows the same base pairing rules as DNA, except that uracil substitutes for thymine. The DNA sequence where RNA polymerase attaches is called the promoter, and the sequence signaling the end of the transcription is called the terminator. The stretch of DNA that is transcribed is called a transcription unit. The RNA polymerase then binds to the transcription factors and the promoter. However, in bacteria, RNA polymerase binds directly to the promoter without the assistance of transcription factors. Promoters signal the transcriptional start point and usually extend several dozen nucleotide pairs upstream from the start point. Transcription factors mediate the binding of the RNA polymerase and the initiation of the transcription. The completed assembly of transcription factors and RNA polymerase II bound to a promoter is called a transcription initiation complex. A promoter called a Tata box is crucial in forming the initiation complex in eukaryotes.
As RNA polymerase moves along the DNA, it untwists the double helix, usually 10 to 20 bases at a time. Transcription progresses at a rate of 40 nucleotides per second in eukaryotes. A gene can be transcribed simultaneously by several RNA polymerases. Nucleotides are then added to the 3' end of the growing RNA molecule. It is important to note that the mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. In eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. Eukaryotic cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA. This is known as RNA processing. before the genetic messages are dispatched to the cytoplasm. During RNA processing, both ends of the primary transcript are usually altered. Also, usually some interior parts of the molecule are cut out, and the other parts spliced together. Each end of a pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide, 5' cap. The 3' end gets a poly A tail. These modifications share several functions. For example, they seem to facilitate the export of mRNA. They protect mRNA from hydrolytic enzymes. They help ribosomes attach to the 5' end.
RNA splicing usually occurs in between the two processes. Complexes of proteins and small RNA molecules called spliceosomes carry out RNA splicing. The RNA component of a spliceosome is called small nuclear RNA, or snRNA. The snRNA joins with protein to form a small nuclear ribonuclear protein, or snRNP. snRNPs and other proteins together form spliceosomes. Alternative RNA splicing is another form of splicing. A single gene is capable of producing more than one different polypeptide by removing different introns from the primary RNA transcript. As previously noted, humans have an estimated 20,000 genes, and these genes are able to produce as many as 100,000 different proteins. Introns and exons are also very important. Eukaryotic genes and their RNA transcripts have long, non-coding stretches of nucleotides between coding regions. These regions are called introns. The other regions, or exons, are called that because they are usually expressed for amino acids. RNA splicing removes the introns, which allows the exons to join together. This creates an RNA molecule with a continuous coding sequence.
Amino acyl tRNA synthetase catalyzes the covalent bonding between the tRNA and the amino acids. There are 20 different amino acyl tRNA synthetases, one for each amino acid. This process requires ATP as an energy source. Initiation factors participate in binding the two ribosomal units to mRNA. The translation process of protein building is helped by an mRNA strand, a large and small ribosomal unit, and then the corresponding amino acids. The first step is for the mRNA to enter the ribosome in order to make the protein. Each triplet, or 3-letter sequence, is used to create one amino acid. A string of amino acids create a protein. These triplets will have a stop and start codon telling the ribosome to begin making a protein and when to stop. The sequence of amino acids create a different protein every time, so one extra or one less amino acid will create a completely different protein.
The first step is for the first triplet to enter into the ribosome. From here, the first codon is used to create an amino acid. This is the start codon, or methionine. This start codon is placed in the P site of the ribosome, and then the next triplet comes into the ribosome. In order for the first codon to be attached to the next one, the second one must be added into the A site, or the third slot. These two are then shifted to the E and P site. From here, the amino acid is released from the E site, and the process is repeated for the next amino acids so they all link together. After they all link together, the protein is released from the ribosome because the stop codon has relayed that message. The mRNA and the protein are released into the cell, and likely another mRNA will enter to the ribosome and make another protein, maybe the same one or another altogether.
A three-dimensional model of the polypeptide chain was constructed using toothpicks to signify a single or double bond, and marshmallows with the colors white to represent carbon, orange for hydrogen, yellow for nitrogen, green for sulfur, and pink for oxygen. A 3D model is now pictured.